Welcome to today's lecture on cell cycle regulation. In this session, we will explore various aspects of cell cycle progression and its regulation, with a particular focus on the role of cyclin-CDK complexes, cell cycle checkpoints, alterations in cell cycle regulation in cancer cells, and therapeutic targeting of cell cycle proteins in cancer treatment. Without further ado, let's dive into our first topic, cyclin-CDK complexes in cell cycle progression. The cell cycle is a highly regulated process that ensures the accurate replication and division of cells. Central to this regulation is the activity of cyclin-dependent kinase, CDK, complexes, which consist of cyclin regulatory subunits and CDK catalytic subunits. These complexes serve as key regulators of cell cycle transitions, governing the timely progression from one phase to another. During G1 phase, cyclin D and CDK4/6 complexes promote the transition from G1 to S phase by phosphorylating key substrates that drive cell cycle progression. Cyclin E and CDK2 complexes, on the other hand, play a crucial role in initiating DNA replication during the S phase. In G2 phase, cyclin A and CDK1 complexes control the progression towards mitosis, ensuring proper chromosomal segregation and cell division. The activity of cyclin-CDK complexes is tightly regulated through various mechanisms, including the timely synthesis and degradation of cyclins, as well as the inhibitory phosphorylation by CDK inhibitors. These regulatory processes ensure that cell cycle transitions occur in a coordinated manner, maintaining genomic integrity and preventing aberrant cell division. Cell cycle checkpoints monitor the genome's integrity during the cell cycle. The G1/S checkpoint ensures accurate DNA replication and repairs any damage. The G2/M checkpoint ensures completion of replication and repairs any damage before mitosis. The spindle assembly checkpoint ensures proper chromosome alignment. These checkpoints prevent mutations in cancer. Cell cycle exit and entry into G0 phase are tightly regulated processes involving factors like PRBA, E2F transcription factors, FOXO family, P27, and P16-INK4A. They control cell proliferation in response to growth signals, preventing uncontrolled growth and maintaining tissue homeostasis. Uncontrolled cell proliferation is a hallmark of cancer, often driven by dysregulation of cell cycle control mechanisms. Cancer cells can acquire various alterations in cell cycle regulation, enabling them to bypass checkpoints, sustain proliferative signaling, and evade growth inhibitory signals. One common alteration observed in cancer cells is the overexpression or amplification of cyclin genes, leading to increased cyclin-CDK activity and enhanced cell cycle progression. Additionally, mutations or deletions in tumor suppressor genes such as P53 and PRBA can disrupt the balance between cell cycle stimulatory and inhibitory signals, promoting uncontrolled cell division. Furthermore, alterations in genes involved in DNA repair pathways can compromise the fidelity of DNA replication and checkpoint activation, allowing cancer cells to accumulate genetic changes and promoting tumor progression. Additionally, dysregulation of signaling pathways, such as the PEEK/AKT pathway and WNT/beta-catenin pathway, can disrupt cell cycle regulation and contribute to cancer development. Understanding these alterations in cell cycle regulation is crucial for developing targeted therapies that specifically exploit the vulnerabilities of cancer cells, providing opportunities for effective cancer treatment. In summary, oncoproteins like RAS, cyclin D, MYC, E2F, and Notch promote cell cycle progression and uncontrolled cell division, leading to tumor growth. Targeting these oncoproteins can potentially provide effective cancer treatments. Tumor suppressor genes, like TP53, BRCA1, and BRCA2, are crucial for preventing cancer development. These genes encode proteins that regulate cell cycle progression, DNA repair, and apoptosis. Loss or mutation of these genes disrupts cell cycle control, leading to uncontrolled cell growth and the accumulation of genetic abnormalities. The G1/S checkpoint is a crucial part of the cell cycle that decides if cells can progress to DNA replication. In cancer, this checkpoint is often disrupted, allowing cells to divide uncontrollably. Proteins like P16-INK4A and CDKs play important roles, and targeting them could be a potential treatment. DNA replication during the S phase of the cell cycle is regulated by DNA replication licensing factors such as ORC, CDC6, and CDT1. These factors assemble pre-RCs at DNA replication origins, marking them for replication initiation. Dysregulation of these factors can lead to genomic instability and cancer development. The P53 pathway is essential for maintaining genetic stability and preventing the growth of cells with damaged DNA. When activated, P53 halts the cell cycle and promotes DNA repair or triggers apoptosis. Mutations in the p53 gene are common in cancer cells, leading to dysregulation of the cell cycle and increased cell survival. Understanding the p53 pathway is important for developing cancer treatment strategies. Epigenetic modifications, like DNA methylation and histone modifications, and non-coding RNAs contribute to cell cycle regulation alongside genetic alterations. These modifications can affect gene expression without changing the DNA sequence, either by silencing or activating genes involved in the cell cycle. Understanding the interplay between genetics and epigenetics can help develop targeted therapies for cancer. The regulation of cell cycle progression involves multiple signaling pathways that respond to external signals and control cell growth and division. Dysregulation of these pathways can cause uncontrolled cell growth and contribute to cancer. Examples of important signaling pathways involved in cell cycle regulation include the Cyclin-D-CDK4/6-INK4 AirBay pathway, the PEAK/AKT/mTOR pathway, and the WNT/beta-catenin pathway. Other pathways, such as Notch, Hedgehog, and NF-kappa B, also play a role in cell cycle regulation and have been linked to cancer. Understanding these pathways can help in developing targeted therapies for cancer treatment. Cell cycle proteins play a crucial role in controlling cell growth and division. Targeting these proteins has shown promise in cancer treatment. CDK inhibitors like palbociclib and abemaciclib halt cell cycle progression in breast cancer. Other inhibitors of mitotic regulators and proteasomes offer potential treatment options. In conclusion, precise cell division relies on intricate cell cycle control. Aberrations in this regulation result in cancer. We explored the functions of cyclin-CDK complexes, cell cycle checkpoints, oncoproteins, tumor suppressor genes, DNA replication factors, the p53 pathway, epigenetic mechanisms, signaling pathways, and potential therapeutic approaches. Appreciating cell cycle regulation is crucial for cancer advancement and treatment.